Hello and welcome, and today I will be discussing transcription and translation. But before I talk about transcription and translation, I want to go over DNA structure as it's very important when looking into these two processes. And one of the most important parts is looking at the bases, as there are four nucleotide bases that can be found in DNA, which is adenine, or A, thymine, T, cytosine, C, and guanine, G. These bases pair up to form rungs of the DNA ladder, A pairing with T, C pairing with G. And these bases are attached to deoxyribose and the phosphate group. And together, they form a double helix. We'll start with transcription, and the first stage of transcription is the initiation, where transcription begins with RNA polymers, an enzyme that catalyzes the synthesis of RNA, binds to a specific sequence of DNA called the promoter. The promoter region is located upstream of the gene and serves as a signal for the start of transcription. We then move into elongation. After binding to the promoter, RNA polymers unwinds the DNA double helix and begins synthesizing RNA by adding nucleotides to the growing RNA strand in a complementary fashion to the DNA template strand. After these DNA templates, termination is started, where we begin to end the process, where transcription ends with the RNA polymers reaches the end of the gene or encounters a terminator sequence, which is a certain sequence made from the DNA bases, and this signals the end of transcription, which at this point the RNA polymers releases the newly synthesized RNA strand and detaches from the DNA template. And once this RNA is synthesized, it undergoes various modifications to become a mature mRNA molecule. These modifications include the addition of a 5' and a poly A tail, as well as the removal of non-coding sequences called introns. And then this mature mRNA molecule is exported from the nucleus and into the cytoplasm, where it can undergo translation to synthesize a new protein. And some important enzymes that help transcription move along, with one being the RNA polymers, as it helps catalyze the synthesis of RNA from a DNA template during transcription. There are several types of RNA polymers that are specific to different types of RNA, but the most common is RNA polymers 2, which synthesizes the messenger RNA, or just mRNA for short. And then there's the helicase, which helped initially unwind that DNA so that RNA polymers and other important enzymes can get in and replicate the DNA into an mRNA strand. And here's a diagram to help show it. As you can see, the DNA has been opened up, allowing for a template strand to attach itself and be replicated using new nucleotides. And it moves from 3' to a 5'. And once this mRNA strand is made, it can move over into the next process, which is translation. And translation starts with initiation, which involves the assembly of the ribosome of the mRNA molecule. The small subunit of the ribosome binds to the mRNA molecule at the 5' untranslated region and scans along with the mRNA until it reaches the start codon, which is AUG. And this is a universal thing that you'll find in L, MR, and mRNA sequences. And the initiator tRNA, which carries the amino acids, binds to the start codon and positions itself in the ribosome's P site. Next comes elongation, where once the ribosome has assembled and the first tRNA is in place, it can begin. The next tRNA molecule which carries an amino acid that is complementary to the codon in the A site of the ribosome, enters the ribosome and binds to the mRNA. The ribosome then catalyzes the formation of a peptide bond between the amino acid carried by the tRNA in the A site and the amino acid carried by the tRNA in the P site, forming a dipeptide. The ribosome then translates the tRNAs, moving the peptide to the P site and making room for the next tRNA in the A site. This will then end with termination, which is the final stage, which occurs when the ribosome encounters a stop codon, which can be UAA, UAG, or UGA, which will be found in the mRNA molecule. When the ribosome reaches a stop codon, there is no more tRNA molecules that recognize it, so a release factor protein binds to the A site of the ribosome, causing the ribosome to release the polypeptide chain, dissociate itself into its two subunits. The polypeptide chain is free to fold into the final three-dimensional structure, and the mRNA molecule can be recycled or degraded, and this ends with that polypeptide chain becoming a protein in the Golgi body. Now, there's many different things that can go wrong during this process called mutations that will occur in DNA, and these mutations can take many different forms, such as the point mutations that will change a single nucleotide in the DNA sequence, which can occur when a change in the DNA sequence does not change the amino acid that is coded for, which will usually have no effect, or next one being when a change in the DNA sequence results in a different amino acid being incorporated into the protein, and depending on the nature, it can have many different effects to no effect at all. And when a change in the DNA sequence creates a premature stop codon, which will stop it before it needs to, which will often ruin the protein it is creating. And then the next type of mutation is a frameshift mutation, which occurs when one or more nucleotides are added or deleted from the DNA sequence, causing a shift in the reading frame of the mRNA, which can be either adding a nucleotide or deleting one. Overall, mutations in DNA could have a range of effects on translation, from no effect to a complete loss of function. The impact of a particular mutation depends on its location in the DNA sequence, the nature of the change, and the context of the affected gene and protein. Some mutations can also have beneficial effects, such as conferring resistance to certain diseases or environmental stresses. Mutations, sometimes it has no effect, sometimes it has a good effect, sometimes it just has a bad effect. And here's a diagram of translation. As you can see, the mRNA strand travels through the ribosome, with the codons being read and a tRNA molecule bringing that anticodon, and along with it, the amino acid, which will then bond into the polypeptide chain, which can then later be released from the ribosome, and where it can then become a protein. In conclusion, and a little summary of transcription and translation, are that there are two primary processes by which genetic information is used to synthesize proteins, which are the building blocks of life. Transcription occurs when RNA polymers binds to a DNA template and synthesizes an RNA molecule that is complementary to the DNA sequence. This RNA molecule, known as a messenger RNA, is then processed and transported out of the nucleus and into the cytoplasm, where it is used as a template for translation. Translation occurs when ribosomes read the mRNA template and synthesize a polypeptide chain or protein, using amino acids as building blocks. This process involves the coordination of several components, including transfer RNA, and can carry specific amino acids to the ribosome, and various translation factors which help to catalyze the formation of peptide bonds between the amino acids. During both transcription and translation, errors can occur that can lead to mutations in the genetic code. These mutations can have a range of effects on protein function, from no effect to complete loss of function. Understanding the mechanisms of transcription and translation is critical for understanding the basic biology of cells and organisms, as well for developing new therapeutics and treatments for genetic diseases. Thanks for watching.